I'm Jen. I'm a postdoc in Kristen Maynard's lab, and I work closely with Leo and his group to do a lot of single cell analyses, and I'm learning a lot of different QC metrics and stuff from them as I'm pretty new to this actually. So, sorry, just looking for my presenter view. So today I'll be presenting on the strategy that our group is taking to investigate and deal with doublets as part of quality control steps for single-nucleus RNA-seq analyses. And this is still sort of an active area of discussion and effort in our group. So here I'm presenting the strategy that we're taking currently. And this is based off of really hard work from Luis, Manisha, and Nick, whose code I've directly adapted and who I've also been learning from and consulting with as I familiarize myself with this strategy to deal with doublets, as well as to just learn the QC pipeline generally. So just wanted to give a shout out to them as I'm very grateful for their help and guidance. So topic of the day, what are doublets? So droplet-based sequencing such as DropSeq or 10x Genomics Chromium uses microfluidic technology to isolate single cells or single nuclei and uniquely barcoded beads into these nanoliter-sized lipid oil droplets, which contain a buffer with all the necessary enzymes and reagents for cell lysis and hybridization of these barcodes to the mRNA fragments. So each of these droplets acts as a separate reaction chamber, and you get doublets when you have more than one cell in one of these droplets. So there's different flavors of doublets. So you can have cells of, for example, the same type. So these are called homotypic doublets, denoted by this gray dashed line. And then you can also have cells of different types in a doublet or heterotypic doublets. And these homotypic doublets are usually not considered as much of an issue since they sort of mimic the transcriptome of a single larger cell of the same cell type and aren't really creating potential hybrid or fake cell types or new transcriptional clusters. So the number of doublets can be influenced by a variety of different experimental variables. So I'm just listing some here. So labs can have different protocols for library prep and upstream wet bench techniques that can influence the number of doublets, or you can be using different versions of this technology that can influence this number. So I know in our groups, we're shifting over from chromium V3 to V4, which might have some additional technical or chemical changes to the technology. Also the number of cells loaded, so the concentration of cells input into your reactions, the cell size, as well as things like cell stickiness. So for example, cells that aggregate more easily, such as immune cells. And our group has added some steps to assess and deal with doublets within our upstream quality control pipeline. So I'll cover this briefly as I delve into the specific doublet strategy we've been taking. So you receive your sequencing reads back, and you need to align them first to a reference genome using software like Cell Ranger. And then we perform a variety of different quality control steps at the cell level. So we identify and exclude any empty droplets. So these are those lipid droplets that don't contain any cells or could potentially just have ambient RNA. And then we also identify doublets using this package called ScDblFinder, which I'll go into greater detail in the next few slides. And then we also identify and exclude any outliers if the cell meets any one of these three criteria. So low total gene counts, low total genes detected, or high percentage mitochondrial reads. And at this stage, we perform the QC and exclude empty droplets and any outliers, but we just identify and label doublets and obtain doublet scores and keep them in the analysis to move forward with and investigate further. So ScDblFinder focuses specifically on identifying heterotypic doublets. So these are doublets that have cells of different cell types. And it's asking essentially, does this real cell in my data set look more like a simulated doublet or more like a normal kind of cell, single cell? And under the hood, the way that it's calculating this, it first simulates artificial doublets. So it will randomly select pairs of real cells from your data set, sum their counts to simulate what a droplet containing two cells would look like, and then adds these artificial doublets back into your original data set. So then now you have a mix of real and artificial cells, or single and doublets. And then dimensionality reduction and clustering by KNN. Graph clustering is performed so that every cell, whether it's real or artificial, has neighbors in PCA space. And then for each of the real cells in your data set, ScDblFinder will ask how many of its neighbors are artificial doublets? Does it sit between clusters? And does it have an unusually high library size? And these measurements are then fed into a classifier which outputs a few like per cell metrics. And I'll just focus on a few here that we assess downstream. So this first one is very important. So it's the ScDblFinder score, which spits out a final doublet score between like 0 to 1, which is essentially the model's estimate of how similar a real cell is to a simulated doublet. And this is mostly data-driven and doesn't really depend on any strong assumptions on things like the expected doublet rate. And then using these ScDblFinder scores, they'll also apply a threshold to label each cell in your data set as a singlet or a doublet. And this is based on a combination of like the score distributions as well as an expected doublet rate from NX Genomics. So within the package, the default is 0.8% per 1,000 cells. So in other words, the like expected doublet rate from the technology informs the decision boundary in this score space. So this is highly dependent on this expected doublet rate, which we talked about earlier, could be influenced by a lot of different like technical experimental variables. So in our approach, we use both the ScDblFinder score as well as these class labels in further downstream analyses to investigate the data, to then like kind of decision make and eliminate doublets based off of our data rather than fully relying on this like class label alone. So just as a reminder here, we're excluding empty droplets and outliers, but keeping the doublets and moving forward. So then with this sort of cleaned up data set here, we perform dimensionality reduction, batch correction, and then we cluster, obtain marker genes for the clusters that we get, and then register them to a reference data set based off of these marker genes to then call our clusters the various different cell types from published data. So this is where we assess these ScDblFinder scores and labels a little bit further to ultimately decide which of these to actually drop. So here we decision make sort of based on how the doublets behave in the clustering. So we are trying to like cluster out as many of them as possible here. And I'll go into a little bit more detail on that. But before I sort of delve into the specifics of this process, I'll just give a few like dataset details of the dataset that I'm presenting here, just focusing on the technical details as they relate specifically to clustering and registration. So this Single-Nucleus RNA-seq dataset comes from the mouse brain, where we tissue punched the habenula brain region, which you can see here. And the habenula is like further subdivided into the medial and lateral habenula subregions. So in our data, we expect a mix of neuron subtypes coming from both of these subregions, as well as glial cell types like astrocytes, microglia, oligodendrocytes, and so forth. And our clusters in our data were registered to this Hashikawa 2020 mouse habenula reference dataset, which includes 6 lateral habenula and 6 medial habenula neuron subtypes, and then 2 astrocyte clusters, 3 oligodendrocyte clusters, and so on. So first, we optimize the parameters for clustering using kind of a combination of K and resolution parameters. So we used for this preliminary clustering Leiden, which is a graph-based clustering method that's commonly used for single-cell analyses. So this K parameter is the number of nearest neighbors that's considered per cell, so it controls how connected your graph-based clustering is. So relatively like smaller K emphasizes more local structure in your data and often produces higher numbers of clusters. And then this resolution parameter controls the granularity of your clustering, so how willing the clustering algorithm is to split your groups apart. So lower resolution typically gives you like fewer clusters. And this is exactly what you see here across a variety of different K and resolution values. So ideally, you want to aim for like a combination of K and resolution parameters that give you a quote like reasonable number of clusters. So we try to aim for less than 50 within our data sets. And though this is a bit of like an arbitrary cutoff, we found that this range has worked well in the past with similar data sets. And this immediately rules out some K and resolution combinations that give you very high numbers of clusters. So then we can focus in on just a subset of this data. And here I'm highlighting the K and resolution combinations that we move forward with for further consideration. So you can notice that these are all under 50. But we also want to keep in mind that within these range of values, having like potentially relatively higher numbers of clusters could help to separate out your doublets more if you're trying to cluster them out. So in the next slide, I'll just be focusing on this subset of the clusters that we decided pass kind of like this metric or thresholding step. And so here I looked further at the median doublet scores for clusters across all of these candidate K resolution combinations that had less than 50 clusters. So each bar is a K resolution combination, and the numbers above the bars indicate the total numbers of clusters in each of these. And every point on the bar plots are the median doublet score for any given cluster within this parameter combination. So you can immediately appreciate here that there are these really stark drop-offs in median doublet scores, indicating that some of these labeled or some of these clusters are likely made up of a lot of originally labeled doublets. And looking across all of these options, I've just highlighted 5 parameter combinations that I moved further with for kind of like further investigation, which maximized the separation of median doublet scores to, for example, eliminate, options like these ones on the right, where you have some more ambiguous clusters that have median doublet scores around like 0.3. So if you were to try and draw a cutoff, you might be like excluding a cluster that has more singlets than doublets. So I tried to basically focus in on the parameter combinations that give you like a greater separation of doublet scores. And then also here I aimed for like 20 to 30 clusters within the resolutions, because if you drop some of the clusters, you're like, for example, on this like 19, 13, or 10 range, you might be left with fewer numbers of clusters that might not be as informative for your data. So then we focused in on these 5 K-res combinations a little bit further. So you can look at each of the K-res combinations. So I'm starting with K20, resolution 0.1, and you can see, like if you sort them, the clusters by the median doublet score, there's this like stark drop off at 0.684 down to clusters that have around 0 median doublet scores. And we see that these first 11 clusters have not only like really high median doublet scores, but also high proportions of like originally labeled doublets. And together, if we were to drop these clusters, they would make up around 15.4% of the total original like labeled doublets that ScDblFinder class gives you. So you can sort of assess your candidate K resolution combinations with this approach. So if we look across our data in a similar fashion and verify that these putative like doublet clusters have high fractions of originally labeled doublets, then we can maximize or try to maximize this number, the total classified doublets that we dropped, which would identify this K25 resolution 0.2 as our best candidate to move forward with based off of these metrics. But then we can also use the actual registration to the reference dataset to calculate a number of different measurements to further assess your 5 or like however many candidate parameter combinations you're dealing with. So here I just want to emphasize that these measures were specific a bit to the goals of our study and could be tweaked a little bit for other studies. So we cared mostly about characterizing habenula neurons over other cell types here. So ideally, with the registration, you want to maximize the number of one-to-one mappings of your dataset's clusters to reference clusters. So this unique non-habenula score gives you a fraction of non-habenula cell types with one or more clusters that map uniquely to the reference. And then this unique habenula score gives you the fraction of habenula cell types that have one or more clusters that map uniquely to habenula subtypes. And you also want to maximize the number of mappings generally to habenula reference clusters. So this shared habenula metric is the fraction of habenula cell types with one or more clusters that map non-uniquely to the reference clusters, but still remain within medial habenula or lateral habenula. So for example, if you have like a cluster 2 that maps to medial habenula 1 and medial habenula 3, it would still count towards this metric. But if that same cluster mapped to like media habenula 1 and lateral habenula 2, it would not count. And the final score can be computed from these three metrics as an average of the three. So in this case, all of the measures are weighted equally, with the habenula-specific metrics together here having more weight than this non-habenula metric. And we considered like any, I guess, like K-resolution combinations that give you more than 10 ambiguous clusters as having a final score of 0. So ambiguous meaning like poor cluster registration values or mapping to multiple different cell types. So based off of these metrics, this K25 resolution 0.2 would be your like best choice if you're just considering like the spatial or cluster registration. And this agrees with the decision based on maximizing the total percent of original doublets dropped. So if you look at the actual like cell numbers for this K-res combination that we want to move forward with, you can appreciate that pretty much all of the cells in the clusters that you would remove were originally called as doublets, and that the clusters that remain are like well sized. You don't really have clusters that are like N of 1 or like N of 5, and you're maximizing the percentage of original labeled doublets that you're dropping. So this is a way to consider both the original calls that ScDblFinder gives you while also looking at your data a little bit more and not throwing away too much of the data. So if you had just gone with the strict cutoff of using singlet versus doublet labels, that removes around like 11% of the data. So here we're trying to keep as much of the precious data as possible by assessing the doublet scores a bit further. So then when we look at the actual registration of our clusters to the reference data set, you can also appreciate that you're not like removing entire reference clusters when you drop the doublets. So we can focus specifically on like 19, 20, 24, and 28, which are removed, which all register to like astrocyte 1 and 2 in the reference. But if you remove these doublet clusters, you still, for example, have cluster 6, which maps very strongly and now cleanly to astrocyte 1 and 2. So just to summarize, in terms of identifying and dealing with doublets, we identify doublets with ScDblFinder and obtain doublet scores at the quality control stage, and then further assess various metrics to like optimize KN resolution parameters by looking at total cluster numbers, drop-offs and median doublet scores, as well as assess how these KN resolution combinations cluster out the doublets. And here we can look at the number of cells that you have in each of the doublet clusters, the fraction of original doublets, while trying to maximize the total like originally classified doublets that you drop with this approach. And then you can assess the 3 scores that I talked about to see how well across your candidate combination of parameters the clusters in your data map to a reference data set. And you use all of this information to drop your doublet clusters, and then with the remaining data, kind of go back to the quality control level and move forward with dimensionality reduction, clustering, and further analyses. So this approach basically sort of like cleans up the doublets in your data set while balancing keeping as much of the data as possible to move forward with. So I just wanted to do another shout out to Luisa Manisha, who trained me on like this quality control pipeline and who I consulted a lot with on like the ScDblFinder scores and how to like deal with them in the clustering. And then Nick for sharing his code for this like assessment of registration to clusters across candidate parameter combinations.